So we looked at arteries and veins, and if we thought about it, there's a lot of similarities, but then so they always are going to have this lining, this endothelium, which is basically what is making up the endothelium of a blood vessel.
Simple squamous.
It's simple squamous epithelium. So that simple squamous epithelium, also called the tunica intima. So sometimes when you're just reading about things about the cardiovascular system, they'll say intima or call it endothelium. It's also what's lining the heart chambers, although when you're talking about the simple squamous epithelium lining the heart chambers, you call it the endocardium instead of the endothelium. When you look at capillaries, capillaries are pretty much pure endothelium. Basically the thinnest kind of wall you can make so you can maximize the diffusion of oxygen and other things across the walls here. Other arteries and veins are going to have this tunica media, which is elastic fibers and smooth muscle, as well as usually some connective tissue on the outside, usually called the adventitia. We talked about veins having these one-way valves in them because the pressure is really low. You want to keep the blood flowing in one direction. There's the massaging of the external muscles. There's that respiratory pump we talked about. Just by breathing in and out, you're changing the pressure in your thoracic cavity, which is helping drive the flow of blood back to the heart. So capillaries, let's talk a little more about capillaries. Well, actually, we talked about what were the two main versions of large arteries we were talking about. There were two kind of classifications. Anybody remember that? Talk about the muscular versus elastic. Pretty sure we were doing that. Like if we, right, like the large arteries, this is not a great graphic. This is supposed to be the heart. This is supposed to be like the aorta or the pulmonary trunk. This is what we were talking about, how your diastolic pressure doesn't drop to zero when your heart is relaxed and refilling. Because during systole, these large elastic arteries, again, like the aorta and the pulmonary trunk, expand and they're very elastic. And during diastole, when the heart is relaxed and refilling, the semilunar valves shut. And as the elastic arteries compress, that's what's generating the pressure. That keeps your blood pressure from dropping to zero. So that's that diastolic pressure is generated by these elastic arteries. Right, the rest of your arteries we usually think of as muscular arteries. And there you just have smooth muscle that's about, again, we talked about that's really important for controlling blood pressure because the diameter of the vessel is going to adjust resistance. Resistance is a huge part of controlling blood pressure. So, anything else interesting here? So let's talk about capillary beds. And this is also going to get into our introduction into the lymphatic system as well. So let's do that. Stop this. No, it's not that. Stop the share. And let me just check in with you. I actually been working really frantically getting the EKG lab going. So I did not look at the video from Monday. And then I've also been talking about this in anatomy. So I want to make sure that I remember what I've already talked about in anatomy, or what have I talked about with you guys? Have we talked about the 3 kinds of capillaries yet?
No.
Okay, so I'm going to introduce the 3 kinds of capillaries.
The very last thing we talked about was the respiratory pump.
Okay, so that was kind of driving the blood flow in the low pressure veins. So that's good, thank you. Okay. So capillaries. So these are, where the business end of the cardiovascular system is. This is the site of exchange between the, the blood and the tissues. And it goes both ways, right? You might be delivering oxygen or delivering nutrients, or you might be picking up wastes and carbon dioxide, or sometimes you're actually picking up products, like in the endocrine glands, you're picking up hormones to distribute, or in the digestive organs, you're picking up nutrients that you're absorbing from your diet. So things are leaving the blood going into the tissues, things are leaving the tissues going into the bloodstream. There are 3 main types of capillaries. And let's do those. So 3 main types. 2 of them are kind of specialty ones that I'll talk about. Actually, let's do the 2 specialty ones first, and then we'll go into the main ones. So this is a specialty type. They're called fenestrated. So fenestrated, it means having windows. So in general, if you have a capillary, it's pretty leaky because, again, it's just made out of simple squamous epithelium. And in general, it has to allow things to move across the walls because the whole job is to exchange things. In a fenestrated capillary, however, they're super leaky. So in addition to the normal ways for things to enter and leave the blood, there's all these little pores or fenestrations. And these are super leaky. So these are going to be found where you either want stuff to be able to leave really easily or enter really easily. These, you've already seen these actually in the choroid plexuses in the brain when we looked at how CSF was created. CSF was created where you had kind of a fenestrated capillary. And a lot of the water and small dissolved salts and stuff are leaking out. That we call that filtration. Kind of think about filtration if you're making coffee through a pour over. You've got the coffee grounds in a filter, then you pour the water over, the water kind of dissolves the caffeine and yummy coffee flavored stuff, but it leaves behind the big things, like the coffee grounds stay in the filter. In filtration, the blood cells and the large proteins stay inside the blood vessel, but the water and very small dissolved solutes can leave. So this is, again, one of the core processes happening, again, when we made CSF in the choroid plexuses. This happens in lots of different places where you're creating these fluids. It's also going to be a key part of what's going on in the kidneys when we do the kidneys in a little bit. So filtration just means fluid in very small dissolved solutes can leave the blood vessel, leave the capillary. Fenestrated capillaries can also be used where you really want to absorb things really efficiently, like in an endocrine gland where the whole job is take in all the hormones so you can then distribute them, or in your digestive organs where you're absorbing nutrients. You want to have them particularly able to, pull things in. So fenestrated capillaries, specialty, super leaky for really kind of abundant, either having stuff leak out or come in. Another specialty type. So another specialty type. They're called sinusoidal capillaries. These are found in the liver, in the spleen, in the bone marrow. So so a sinus means kind of an open space. It's not even really a capillary like you normally think about it anymore. So let's say, they're usually really windy. They're kind of places where, you even have breaks in the walls. So let's say there's so you can have surrounding cells kind of reach in actually and process the blood. So this is places where the blood is getting filtered and cleaned. Like in your liver and your spleen, you actually have white blood cells reaching in and recycling worn out red blood cells and things like that. So the blood is percolating through slowly. The surrounding cells have easy access to the blood in there to help kind of process and clean it. So again, the only places that I know that these are important are in the liver and the spleen and the bone marrow. We'll talk more about the liver when we do the digestive system, and we'll talk more about the spleen today. All right, so that is going to bring us to the most common kind of capillary. Where is my text? Okay, most common type of, most common type, they're called continuous capillaries. So these are the ones you normally think of, like taking the blood in, delivering the oxygen and the nutrients, picking up the CO2 and going back to the heart for more. So you're basically going to have some arterial coming in. Again, that's kind of the smallest little branch that are of the kind of arterial side. Eventually branch into the actual little capillaries. We call the capillary bed. And then the capillary bed will drain into a venule. So this here is my capillary bed. Things about capillary beds. One thing is capillaries. The diameter of a capillary is around 10 microns. 10 millionths of a meter. The diameter of a blood cell, a red blood cell, is only 8 microns. So this means that red blood cells are basically going through single file. So that's going to maximize the ability to diffuse oxygen, right? You have the red blood cells, which are basically the size of the diameter, which means they're right near the wall, which is going to minimize the distance oxygen has to diffuse to get from the blood into the tissue. So the diameter of the vessel is about the same as the diameter of a blood cell. So RBCs are going through one at a time, single file. Other things that are important here. There are little precapillary sphincters, smooth muscle that will regulate the entrance to the capillary bed. This would be better if I just type it. So precapillary sphincter, smooth muscle, does a couple of things. One thing is this precapillary sphincter can determine whether or not the blood enters the capillary bed at all, right? Because your tissue perfusion is going to depend on how metabolically active the cells are in that region at the moment, right? So let's say this is in your skeletal muscle. Let's say this is like, skeletal muscle here. Right now, you are just sitting in front of your computer. You are not doing a lot with a lot of your muscles. Your muscles don't need a lot of oxygen to just sit on your butt right now. So right now, when you're at rest, about 90% of the capillary beds in your muscles are actually not being perfused, are just closed off. So right now, only 10% of the capillary beds in your muscles are getting blood going through them. The little precapillary sphincters can just close down. If you actually start running around and moving and you need more blood going in to deliver more oxygen, you can start perfusing more and more of the capillary beds. Another thing that these little precapillary sphincters do is even when you are perfusing the capillary bed, it does it in this kind of jolting kind of way. They open and then they close, and they open and they close, and they open and close. So it kind of, they think it's to kind of keep kind of jolting so the blood doesn't accidentally like get stuck or back up. So you basically open it, it's starting to go, and then it closes and then it opens. So it's constantly like kind of giving it a little shake to make sure things keep moving along. If a capillary bed is closed, you still need some way for the blood to have some minimal flow so it doesn't just get stuck on this side. So there are these bypasses, which I'll just draw in right here. This thing here, this is called a meta-arterial. Right, so even if the capillary bed is closed, there's still going to be some minimal flow from one side to another. So that is your classic continuous capillary or continuous capillary bed. What I'm going to do now is introduce the lymphatic system. We are going to come back to the blood proper and the blood cells, but let's, while we're here, let's just talk about some aspects of this capillary bed and why we're going to have to bring in the lymphatic system. So I'm going to get rid of this. All right, so we have these are just supposed to be cells of your tissue. And then the space in between your cells, that's that extracellular fluid. We often call it interstitial fluid. Interstices are just, interstices are the little spaces between. So the interstitial fluid is just the fluid that is in the spaces around the cells. So when the blood enters the capillary bed now, typically the blood pressure, like the blood pressure coming out of the ventricle, the left ventricle, we talked about the mean arterial pressure. In fact, you should all remember how do I calculate it? If my blood pressure was 120/80, what is the mean arterial pressure for that? How do I calculate mean arterial pressure? Does anybody remember? Nobody? I keep mentioning mean arterial pressure, and I know you calculated it for your lab. This was kind of that average, somewhere in between, but weighted down towards the diastolic. It was basically diastolic plus 1/3 of the systolic minus the diastolic. And here, if diastolic is 80, times 1/3 of 120 minus 80, that's going to be 80 plus 1/3 of 40. 1/3 of 40. This is actually like 30 plus 10, so that makes it easy. This is 10 plus 3 1/3. So this is going to be 93.3. Right, so that's mean. You should know mean arterial pressure, what it is, how to calculate it. So if somebody's blood pressure is 120/80, that means their mean arterial pressure is 93.3 millimeters of mercury. Right, because your book is talking about mean arterial pressure all the time, so you should know what that is. So mean arterial pressure coming out of the ventricles is up above 90. But as you get into the smaller and smaller and smaller and smaller little branches of the cardiovascular tree, you start having less and less pressure. So by the time you are entering into the capillary beds at this arterial, the pressure is down to about 40 millimeters of mercury. So that's the pressure as you're entering into this arterial. After you have spread out into these super, super tiny little capillaries, on the other side, you're down to about 20 millimeters of mercury. Right, that's why you need all of those tricks to try to keep the blood getting all the way back to the heart. If you only have 20 millimeters of mercury left, that's not a lot of driving force to take you all the way back up the vena cava, back into the right atrium. So here's our setup. The fact is, though, there's quite a bit of this kind of hydrostatic pressure. It says low, but it's still pressure. So we've got this pressure, and we know that capillaries, even continuous capillaries, they're leaky. They have to be leaky to some degree, or else stuff can't enter and leave the bloodstream. And so as the blood enters into the capillary bed, water leaves into the tissue. So this is water as well as other kind of small dissolved solutes. So as we enter into the capillary bed, you know, water leaves. It's mainly due to kind of the hydrostatic pressure. You've got blood entering in under pressure into this kind of tube that has some leak to it, so we have water leaking out. And it's going into this interstitial space, the space between the cells. As we get to the other side of the capillary bed, conditions change a little though. The pressure is lower on this on the far side. Plus, what is happening to the osmolarity inside of the blood as we start losing water? What happens to the osmolarity of something if you start losing water? Taking away water.
Goes up.
It's going to go up. Now, all of a sudden, there's more dissolved stuff compared to the amount of water. So we're actually going to have a lot of this water returning back in on the far side here. And that's going to be both because we're going to have osmotic pressure here. You know, as well as so on the far side of the capillary bed, you are going to end up having the water returning. So this is something that's happening in your capillary beds all the time. As blood enters the capillary bed, water and dissolved stuff gets out into the tissue spaces, but on the far side, most of it comes back in. However, not all of it. So there's going to be a net loss. Let me do this black. Black. Like around 3 liters per day. So every day there's a net loss of about 3 liters out of your bloodstream just into this interstitial space, the space between the cells. Obviously, that's something we got to deal with, both because you don't want your blood to turn to sludge. If you just lost the water and didn't get it back in, you're just going to end up with mud in your blood vessels. Also, you're going to have all this water just coming into the spaces between your tissues, and it's all going to get all swollen with extra fluid in between that it shouldn't have. So we need to have a system to collect that leaked fluid and dump it back into the closed circulatory system. And that's going to be one of the important roles of the lymphatic system. So hold on one moment. Technical deal I need to deal with here so I can keep track of things. All right, continuing back. So this 3 liters per day, we need to somehow collect it and return it back into the closed circulatory system. And we'll see also in the process, we're going to filter it as well, kind of clean it up. So that is going to be our introduction to the lymphatic system. This is going to be done by these little lymph capillaries. So let's take a look at those. So the basic idea here is, in addition to your capillaries, your blood vessels, you're going to have all these little lymphatic vessels. For whatever reason, People have the convention of drawing lymphatic vessels in green. And I will continue that convention. It just makes it easier to kind of draw this and know what you're seeing. So I'm going to draw these vessels right now in green. And these are going to be my little lymph capillaries. So lymphatic capillaries. Whoops. Let me. So these are basically little capillaries. They're kind of like a regular capillary, although there's a few differences. One difference is obviously they just start in the middle of nowhere. Ready? Normal capillaries, there's an entrance to the capillary, there's an exit to the capillary. These lymphatic capillaries are just blind little alleys that are going to be absorbing the fluid and taking it away. So they don't have something feeding into them. They just kind of have these little blind fingers coming into here. Another difference of a lymph capillary is the little endothelial cells that make up their walls have this ability to kind of flap open. So endothelial cells can kind of flap open. Which means that they can absorb bigger things, right? In fact, these are kind of, kind of anchored in the connective tissue in your body. And just as you move around, these things kind of get opened and closed. And as they open, they kind of suction in whatever is floating around in here. They suction in the water, they suction in proteins, they can even suction in things as big as cells, because these flaps are big enough that you can actually suck in a cell even. We'll see that is actually going to be relevant when we talk about, why do people take out lymph nodes when people have cancer and things like that. So these lymph capillaries are basically absorbing the water that's been leaked out. They can also absorb any other kinds of things that are out there, again, proteins, and again, things as big as cells. So lymph capillaries everywhere. Those lymph capillaries then they start coming into larger and larger lymphatic ducts or lymph vessels. Lymph vessels, because there is basically no pump, right? There's no heart for this. So they're going to have to rely on the same kind of tricks that the veins used to keep the flow going. These are all going to have little one-way valves in them. They are going to rely on the pulsing of things outside of them to kind of milk them, right? So muscles and even blood vessels. I mean, these things often get kind of bundled along with arteries. And as the arteries are kind of pulsing outside of them, that's helping milk the lymph and keep it moving. So the lymph, and the definition of lymph is just the fluid and dissolved stuff that gets absorbed into the lymphatic vessels. That's all lymph is. Anything that ends up getting absorbed in here, again, the water and proteins and whatever else that's just lymph. Lymph is just what it's called once it's traveling in the lymphatic vessels. As these lymphatic vessels go, they percolate through these little things called lymph nodes, which we'll talk about in more detail, but they're basically things that help filter the lymph. So the lymph node, it has white blood cells that help do immune system. We'll talk more about the lymph nodes in just a few moments, but the basic idea is they can filter. As this lymph is percolating, they can filter it, kind of clean it up. And ultimately, where is all of this going to go? So we've got
Back into the closed circulatory system.
Exactly. We have the lymph coming in, going through lymph nodes. There's actually tons of lymph nodes. It's not just. So ultimately, this is all going, and we're going to have, here's the heart. Here's like the vena cava returning back to the heart. Here's like the brachiocephalic veins. All of these ultimately just dump. And there's another one coming in over on this side. So basically, this ultimately just dumps back into venous circulation. So all that fluid that's being collected all over the body, filtered by the lymph nodes, and ultimately now just dumped back and ends up in the vena cava and is now back just in your closed circulatory system. So it's returned back into general circulation. Does that make sense? So that is that. Now what I want to talk briefly about is what is going on in a lymph node. So before I can talk about what's going on in a lymph node, we need to bring up the idea of lymphatic tissue. Also called lymphoid tissue. You know, it's basically a lot of these white blood cells involved in your immune system. This is going to be like lymphocytes, which are kind of B cells and T cells. We're going to talk about this so much more when we do the immune system. We're going to learn about B lymphocytes maturing in the bone marrow and T lymphocytes maturing in the thymus and how they differ and how they get activated and all sorts of stuff. But for right now, let's just say they are part of your immune system and they're helping keep you safe. Macrophages are another kind of important white blood cell that are going to be here. These, big eaters. These are things that can eat things, take things out. They can actually activate lymphocytes if they find something interesting that they think we need to make an immune response against. So we have these different kinds of white blood cells. These are all white blood cells. They're kind of anchored in just this framework of little reticular fibers. There's a structure, you don't have to recognize it for this class. You often get these little, especially the B cells, in these big nodules. We call like a lymphoid nodule. Also called a follicle. This is where you often have the B cells. And again, we're going to talk about B cells in more detail. B cells are actually the cells that make antibodies. And we actually are going to talk a little bit about antibodies when we do our blood lab today. But the basic idea is You've got this lymphoid tissue. It's got all these different things that are part of your immune system, and it's located in a variety of different places. So I'm kind of introducing the idea of this lymphoid tissue, and now I'm going to look at it in context. And the reason why I'm giving you lymphoid nodules is just because when I draw the pictures, I'm often going to draw it with these little circles around. So I'm just referring to, oh, it often has this kind of image of these little circles kind of thing. So let's go back to our lymph node. A lymph node, it's just this little thing, and it's got a whole bunch of lymphatic tissue in it. It's also got like connective tissue capsule. It's a little bitty organ. It's about the size of a little bean. Think about a little pinto bean or something. It's about the size of a bean. It's got a little connective tissue capsule around it, and it's got a bunch of this lymphoid tissue, these white blood cells that help keep you safe. And the lymph is percolating through it, getting cleaned up, and then continuing on its way. The lymph nodes are all over, but there are three main clusters that people talk about that I should mention. So there are cervical, so clusters of lymph nodes. Cervical, meaning your neck. Inguinal, meaning your groin. And axillary, meaning under your arm. The reason why this is useful to know is, if somebody is fighting some infection, let's say you've got some like, sore throat, or so you've got some infection in your throat. Often people will start getting swollen glands. When people talk about swollen glands, it's actually the lymph nodes that are actually getting enlarged. So these follicles I was talking about, if you have some infection, active infection, you're going to start having a whole bunch of B cells dividing to make more and more antibodies. And as that happens, you're going to get more of those follicles. This thing is going to enlarge, and you're going to actually have the lymph nodes enlarge. And you can, that's the swollen glands. The swollen glands is if the white blood cells in the lymph node are multiplying and enlarging the lymph node. So that's one place we find this lymphoid tissue, is inside the lymph nodes. Another place we find this lymphoid tissue is in the spleen. So let me talk about the spleen while we're here. Oh, think ahead. This was horrible. Hold on, let me undo that. The spleen. So the spleen is this interesting organ. Like if this is you, here's your abdominal cavity, here's your chest cavity, here's your diaphragm, here's your heart. The spleen is located kind of right up here. It's right in the upper left-hand corner of your abdominal cavity, up against your diaphragm. You've got a big splenic artery entering in. And you have splenic vein leaving. And the spleen, you can kind of think of a lot like a lymph node, except instead of filtering lymph, it's filtering the blood. So and I should say filters blood, flowing through sinusoidal capillaries. So basically, you've got these clusters of white blood cells, lymphoid tissue, and then you have blood kind of flowing in and seeping around. And as it's going through, the white blood cells can process and clean up the blood. We're going to talk about red blood cells. Red blood cells only live 3 to 4 months. They don't live that long. This is one of the places where worn out red blood cells are salvaged and recycled. Sometimes people call this like the RBC Graveyard. Because it's one of the places where the red blood cells go to die. This is also a place where you have since blood is kind of just percolating in there, there's a lot of kind of excess blood that's just kind of in there. So if you're hemorrhaging, you're bleeding really bad, the spleen can actually spasm to push more blood into your circulation to bring your blood levels back up a bit. So it can do that. The spleen is not essential. Like if you lose your spleen, you'll still be all right, because this job of kind of filtering and processing the blood is also done by the liver. The liver also has sinusoidal capillaries, also has white blood cells in cleaning things out. So if you're, so sometimes people, a bad accident, if I had a student whose spleen exploded in a car accident and she had no spleen anymore and she was okay. Because the liver can take over the job of the spleen. I think there's some issues. I think a lot of times the platelets, the clotting factors tend to get stored in the spleen and stuff. But in general, the spleen is something you can live without. But again, spleen is different from a lymph node because it's filters blood. Like if you're thinking about what is it about the spleen that is different from like a lymph node, it's filtering blood. Lymph nodes are filtering lymph percolating through them. So the spleen. That lymphoid tissue is also located in other places around your body. So we often talk about malt. Sounds like something you get at the soda fountain, but no. Malt is mucosa associated lymphatic tissue. So that lymphoid tissue is just located all over. Mucosa is just any kind of membrane coating tubes that are continuous with the outside. So for instance, let me unblock. Your digestive tract. Your digestive tract starts here, goes down, there's your stomach, there's your intestines, there's your anus. This is the lining of this whole digestive tract, we call that a mucosa. This is a place where things can get in your body. It's a vulnerable boundary. So you have this lymphoid tissue, which is right on the other side of the wall. So like right in around your throat, under your tongue, on your soft palate, on the back of your throat, you've got clusters of lymphatic tissue. What do we call the specific clusters of lymphatic tissue that are ringing around your throat there? They have a special name. You get them sometimes when you're a kid, people get them taken out. Then they get to eat ice cream.
The tonsils or adenoids?
Yep. So the tonsils are just an example of this. They are, and the adenoids are one of the types of the tonsils. The tonsils, we're not going to go, you don't need to know this different types for this class. There's the adenoids or pharyngeal tonsils, the lingual tonsils, the palatine tonsils. But those are just collections of lymphatic tissue that are kind of just under the epithelium surrounding your throat here. You've got more and more just along as you go along. You know, in anatomy, you learn about the Peyer's patches and things. You know, so but they're all over. They're also on the outside of your trachea. Your trachea is another mucosa, another kind of wet epithelial membrane that's exposed to the outside world. So that's all MALT is. So this lymphatic tissue is basically on the other side of these membranes to try to keep you safe from things getting across and getting into your body. So that's all I'm going to talk about that. And I think that's all I'm going to really talk about lymphatic tissue, although I am going to do a quick little thing here. All right, we got back to here. So what happens if the lymphatics are not working well? What happens if something is keeping the lymphatics from doing their job?
There's swelling.
Oh, say it again.
There's swelling.
You're going to get swelling. So you're going to get an excess of fluid building up in this interstitial space. You can also get it if there's just so much leakage that you can't keep up with it. So when you have excess interstitial fluid, that's when you get into what's called edema. So edema is this excess of interstitial fluid. You can get it actually in, if you have peripheral congestion and congestive heart failure, where you have this kind of back pressure on the systemic side of the cardiovascular system, that's going to mean more fluid than should is leaking out, right? If we're going back to this picture, if this is, you know, if we can't take this up on this side, then we're going to have more pressure on this, more fluids leaking. So peripheral congestion, you're going to have edema. Or if something is causing the lymph capillaries to not do their job, either because you've had to remove a bunch of lymphatics because of like someone's got cancer, or you've got some parasitic worm that's blocking them up, that's going to cause edema. And actually, let me really briefly talk about why do you take out lymph vessels in cancer? So what is cancer?
Normal cell growth.
Yeah, so cancer is when you have some cell that, or some cells that are dividing uncontrollably and creating these tumors. And, you know, if they just stay in place, it's bad enough because they're starting to push on surrounding structures. They're diverting your body's resources and using up your resources rather than letting your cells that need it use those resources. But what gets even worse is if part of it breaks off and then starts seeding new little tumors. So that's when a cancer is metastasizing. That's if the cancer breaks off and has new little stem cells that start new little tumors. When cancers metastasize, because those lymphatic vessels, I talked about the capillaries have the little flaps where they can bring in entire cells. When a cancer metastasizes, often those little metastatic cancer cells get sucked into the lymphatic circulation. And then they end up landing in a lymph node and they lodge there, and then they start making secondary tumors in your lymph nodes. So when someone has cancer, like if someone has breast cancer, they don't just usually take out the tumor. They usually also take out all of the nearby like axillary lymph nodes as well, because that is going to ensure that if there are any secondary little tumors that have started growing in the lymph nodes, you've got those as well. So it's not uncommon to take out surrounding lymphatics as well as just taking out the main tumor. But again, that is going to cause that situation I was just talking about, where you're going to have lots of swelling in that region because now you don't have the normal drainage. Let me show you a few images that are worth showing. Hold on one sec here. Doobie-doo. So one of the things that's interesting about how you know that somebody has edema rather than just kind of fat is that when you like press on them, if someone was just kind of had adipose there, whoops, and you pushed on the fat, it would just spring back. But when somebody has edema, then when you push on that, you kind of basically displace the fluid, displace the water, and then when you let go, that hole just stays there. And that is one of the real defining qualities that you see, whoops, in edema. Let me see if there's other better pictures of it. Yeah, this idea that if you just push on it, it will stay down. And it definitely works. I've done it on somebody and it's just kind of weird. It's like memory foam. You basically push and then it stays down, but then it slowly kind of comes back as the fluid kind of re-equilibrates in the interstitial spaces. Lymphedema, you can get these horrible things. If you have impaired drainage of the lymph, you just start getting the tissue. Whoop, I didn't mean to do that. I can show you, just totally filled with fluid, right? This is just way too much fluid inside the tissues here, and it's inadequate drainage. And so edema is characterized by all of this swelling like this. Some diseases, there's this tropical disease where this little worm gets in and blocks your lymph vessels. And I think it causes scarring that blocks the lymph vessels. So it's called elephantiasis or filariasis. And again, you get, let's get some better pictures. This is somebody's legs that have just been filling with fluid without having the drainage because the lymph capillaries are all clogged up. Here, this again, it's pretty horrifying. Again, look at you can see his legs are just, I think he actually might be pulling out, I know he's probably washing himself there. So anyway, just this idea of how important is it that your lymphatics are doing their job and draining your tissues. It's very important. If they don't, then you start just getting all your tissues all swelling up. Worst case, you get stuff like this. So let us stop sharing that. So that is what we're going to talk about for the lymphatics. We are going to spend way more time talking about lymphocytes and macrophages and the details of the immune system. But for right now, don't worry about them beyond having them being, important cells for our immune system. Your next exam is not going to deal with the details of immune cells. Your exam 4 at the end of the semester, that will be in detail about what is a B versus a T lymphocyte. But for right now, don't worry about it. So let's talk about blood. Actually, wonder if we can, let me see if I can find this. Hold on 1 second. Actually, no, I'm not going to do. There's something really fun, but I'm going to just stay focused here. Blood. It's going to be the focus of our lab today. And so I'm going to kind of introduce it. It's, what kind of tissue is it? This is, you don't really need to know for this class, but it's always kind of non-intuitive to think about blood as a tissue. What kind of tissue is blood?
Connective tissue.
It's a connective tissue because it's a bunch of cells that are widely spaced with this stuff, this matrix, in this case a fluid matrix called plasma in between. So when we talk about blood, we need to talk about the fluid part, which is the plasma, which is typically about 55% of the overall volume. And then what we call the formed elements, which are mainly cells. And that's usually about 45%. This ratio of formed elements to plasma, this 45% here, that is going to be the hematocrit. We're going to look at that in more detail. So let me just. Hematocrit. To get the hematocrit, it's going to be really easy. You're going to take a tube. You're going to fill it with blood. And again, normally the blood is mixed up with the plasma and cells, and it's hard to tell which is which. What technique have we used in lab right now that can pull all the cells down to the bottom?
Centrifuge.
The centrifuge, exactly. So you just take this little tube of your blood and now it's just pure blood. It's cells and plasma. You centrifuge. And when you're done centrifuging, the blood cells are all packed on the bottom. The plasma is up on the top. And then you can just measure. This is what, if we were going to be doing it for real in lab, you would actually do this with your own blood, and then you would just measure what is this total distance You'd measure what is the height of your packed cells, and your hematocrit is just going to be, X over total height times 100. So that's all hematocrit is. It's just this ratio, how much cells to overall plasma. For some, on average, I wrote on the other page, on average, it's around 45, but it changes. Normal ranges anywhere from like high 30s to low 50s. So it's different in men versus women as well. We'll talk about that when we get to lab. So hematocrit, we will talk about this in more detail in lab, but just to kind of introduce this idea. So plasma versus formed elements. Let's start by talking about the plasma. And then we'll get to the formed elements. So plasma is, what's it mostly? It's mostly water. The normal number they use is 92% water. And then other stuff. Obviously, dissolved things, dissolved nutrients and wastes like sugars. Obviously, you're just sending sugar. So, but those don't take a lot of volume. So like if we look at the total amount of dissolved nutrients and wastes and gases, it adds about another 1% of the overall volume there. Nutrients, sugars. There's also going to be hormones. You're going to be picking up urea to get rid of. We'll see oxygen doesn't really dissolve well in the plasma, but carbon dioxide does. When we talk about the transport of carbon dioxide, like 10% of it is just dissolved in your plasma, like, kind of like soda water. Exactly like soda water. So 1% dissolved nutrients and waste, but then that's going to leave us with 7% plasma proteins. So let's talk about the plasma proteins, and I'm going to get rid of the formed elements side here for now, just so I give myself more room to play. The plasma proteins. These are big proteins. They're inside your blood. They stay in your blood. Even like when you have those fenestrated capillaries, these plasma proteins are big enough that they don't leak out through the fenestrations. They tend to stay in. They play lots of different roles. Let's talk about one of the most common of all of these is called albumin. I think it's like maybe 60% of all the plasma protein. One important role that this is playing is just to help maintain osmotic balance between your plasma and the surrounding cells and tissues. Right? If you just had pure water for plasma, you know where that goes, right? Distilled water is super hypotonic, which means water is going to enter into the surrounding cells and tissues and stuff. So you need to have an osmotically balanced fluid environment, or in order to keep osmosis from happening in a way that you don't want it to. In fact, like when you think about like one of the classic these pictures of people starving, but you see them with these big bloated bellies, and it's like, wait, why do they look all bloated if they're starving? Like one of the important types of starvation is kwashiorkor, where you don't get all the amino acids you need from your diet. Like if you just eat grain, you're not going to get all the amino acids that you need to make this protein. And if you don't make this protein, then your blood is not able to keep all the fluid where it belongs, and it just enters into the tissues, and you end up getting all bloated looking and stuff. So osmotic balance. Albumin, as well as others, also act as carriers to help move things through the blood that normally wouldn't dissolve. Like there's a lot of lipophilic things, non-polar things like steroid hormones and vitamins and things like that, fat lipid-based vitamins that don't dissolve in water. They're not going to just dissolve in your plasma. So albumin, there's also alpha globulins. There's beta globulins. These can act as carriers, meaning they bind to things that normally would not dissolve, but then as a complex, they will dissolve and move through your body. There are lots of other carriers as well. There's specific carriers for specific hormones. Often, like, if you're talking about testosterone or estrogen in the bloodstream, you talk about bound versus free, because like those are steroid hormones. Testosterone does not dissolve in water. So testosterone is going to be bound onto a carrier to actually move through the bloodstream. But when it's bound to the carrier, it's not active, actually binding to receptors. So then we talk about the free amount of the hormone that's actually not complex and is actually able to bind to receptors and all that kind of stuff. So carriers, important to move things through the blood that won't dissolve normally. We're going to also see fats are often moved through the blood as lipoproteins, right? Lipoproteins, where you have, protein, lipid. So lipoprotein, that's another way you get non-polar things transported through the bloodstream, is by making a complex have an outer coating that actually is hydrophilic and put the hydrophobic thing inside the storage in the middle. We'll talk about a variety of these lipoproteins later. There are high density lipoproteins, low density lipoproteins involved in moving fats in and out of your system. There are chylomicrons, which we're going to see, that are important for absorbing the fats from your digestive system. So we'll get back to these guys. There are lots of other proteins. Angiotensinogen, your old pal from the whole renin-angiotensin mechanism. That was a plasma protein just sitting in your blood. We're going to be talking about blood clotting in a little bit. That is going to be mediated by like over 20 different plasma proteins that are just sitting dissolved, ready to go in your plasma. When we get to the immune system in more detail, we're going to see the complement system. There's all sorts of plasma proteins doing different jobs. Some of them are carriers. Some of them are involved in blood clotting. Some are involved in body defenses. Some are involved in blood pressure. So they're doing lots of different things. So plasma proteins, again, we'll be meeting more of them. They are made in the liver. We'll talk about the liver when we do the digestive system. It does lots of different jobs, but one of its many jobs is make plasma proteins. There's one extra plasma protein that is not made by the liver, and so I'm going to introduce that, and then we're going to take our break. So let me go on another place. One other plasma protein, and it's not made by the liver. Good God. Rather made by lymphocytes. And that is what we can either call gamma globulins. This is gamma. They're also called immunoglobulins. They're also called antibodies. We're going to talk way more about antibodies as well as lymphocytes that make them and all that when we do the body defenses. But for right now, you need to understand the basics of the antibodies because it's actually going to be a core part of how we're going to do blood typing in our lab today. So what is an antibody? It's a protein. It's kind of this complicated protein. It's got different parts. It's got, we call it, there's heavy chains and light chains. So this is my antibody protein here. It's got a constant region. And it's got a variable region. So they all have the same basic kind of base. But then there is this variable region where they can be different. And to talk about what that means, we have to kind of talk about an antigen. So antibody is the plasma protein. An antigen is the thing that the antibody recognizes. So this variable region recognizes and binds to an antigenic determinant. Recognizes and binds to the antigen. So let's talk about antigen. Antigen, it comes from antibody generating. So antigen can be anything with a particular shape. You know, let's say I've got like my, I don't know, some virus or bacteria that's like, let's give it, we can even give it a little crown. It can be coronavirus. You know, with crazy claws that go dig into your surfactant cells in your lungs and make you not able to breathe. So let's say that is, coronavirus can be an antigen. That's what we're hoping to come up with, some way to stimulate our body to create antibodies for this thing. It's got very distinctive shapes. You know, I made it as a cartoon here, but in reality, there are very distinctive shapes on like the protein coat that actually make up a virus or make up the outside of a bacterium or make up anything. There's distinctive kind of shapes. And the antibody has those variable regions that can recognize a particular shape. So like I could have an antibody where it actually would match that horn over there. But you can have an antibody that can then recognize and bind to that exact antigen. Any particular antigen can often have more than one spot that you can use to make the antibody. So each of these sites we call like an antigenic determinant site. Like I could use this area as my antigenic determinant. So I could have an antibody actually that could also recognize this same little beastie just by recognizing a different location on it. But the basic idea of an antibody is it has this variable region that is specifically able to recognize a very specific antigen. So they're very specific. So they are only going to recognize a particular antigen. And again, when we do the immune system, we're going to talk way more about antibodies and how they're used and what they do in the bigger picture. We'll talk more about the lymphocytes that make them. But for right now, just know if you have an antibody that is particular for a particular antigen, it will bind to it. When it binds to it, it does a variety of things, but one of the things it does is agglutinate. It grabs them together. Because there's two of these regions on each. Like if let's say this was my little thing here, and I had my antibody come in and bind onto that, but it also has one it can grab onto this one. But then another antibody comes in here and binds onto this one, but it also has another one over here, so it binds onto another one. So you start getting what's called agglutination. Basically, the antibodies start connecting all the antigens together and clumping them all up. Right, so these are my little antibodies. So this is going to be the key to actually blood typing. When we're going to do our blood typing, we are going to have specific antibodies that will look at just like the A antigen or the B antigen, and we'll add them to the blood. And if we see agglutination, then we know your blood has that antigen on it. So that is where we're going to stop for now. Are there any questions about all this? More about hemostasis, blood clotting. So let me do that. So this is kind of a little bit of lecture material as well, but it's also going to be critical information for you to understand today's lab. This would be kind of continuing about talking about the blood. So let's take a 5 or 10 minute detour and talk about hemostasis. And then we will return and talk about the 4 different parts of today's lab and actually do them. Hemostasis. So the basic idea is you've got your blood vessels and they're constantly getting little. Sometimes you get big tears, you know, it's like, ooh, in fact, normally in this lab, you would stick yourself with a pin and you rip a hole in the wall and blood starts oozing out. But just in general moving around or getting bumped or this or that, especially the little fine blood vessels are constantly tearing and ripping and blood is leaking out. And so you need to have this mechanism in constant action that stops the bleeding and repairs the damage. So blood stasis stopping. So this is basically the system in place that stops the bleeding and repairs the damage. And at the core of this, you have to remember you have a blood vessel lined with endothelium. And then we are going to introduce the first of the formed elements for you all. So you're going to meet the platelets. Sometimes they're called thrombocytes, but I really don't like that word because cyte implies it's a cell and it's not actually a cell. Platelets, ultimately, what happens is you have like some big specialized cell in the bone marrow called a megakaryocyte. So this is this weird big cell is called megakaryocyte because it's got a really big nucleus. And the way it does it is it makes little granules, well, granule-filled packets. So it makes stuff, fills some little thing and pinches it off. So the platelet is actually just a little vesicle filled with granules. So I said different stuff. We'll talk about some of the things that are in the granules. So basically in your bone marrow, these megakaryocytes are busy pinching off these little packets, which are then floating in your plasma. So these are just normally floating through your bloodstream. They're not really cells. Again, they're just like little packets of stuff. And as long as all they see is endothelium, they just stay intact as themselves. So platelets are just waiting for their job, but normally they're just flowing in your blood like your blood cells are flowing. However, if there is a rip in the blood vessel, then all of a sudden they see something that is not endothelium. As long as there's no rip, they're never going to see anything but this endothelium. These are going to be triggered by exposure to collagen. It's one of the main proteins in your connective tissue. So I'm going to add, so if platelet is exposed to collagen, so again, collagen, structural material, it would be like you walked into the room and instead of seeing paint and wallpaper, all of a sudden you saw 2x4s and pipes and, conduits and, wiring. It's like, oh my God, I'm looking inside the wall. I shouldn't see that. There's been some damage to the wall. So the platelet exposed to collagen, that's only going to happen if there is some rip, some damage in the wall. So, and that is now going to turn on a whole sequence of events. So let's talk about the sequence of events that are all part of hemostasis. The first step, these platelets release, of all things, serotonin. So serotonin, now we think about it in your brain and like the thing involved with antidepressants or this or that. But the first place people learned about serotonin was, oh, it's an important molecule in, hemostasis. This idea that it's an important neurotransmitter that's much more modern. So, and again, the same thing plays different roles, right? I can have a pen and I can use it as a writing instrument, or I can use it as a murder weapon and jam it through somebody's heart or something, right? The same molecule plays very different roles in different situations. So serotonin in this situation is just signaling, triggers vascular spasms. So meaning the smooth muscle in the vessels is going to start contracting. So why is that going to be useful? How is that going to be a useful thing to start triggering vascular spasms if there's a tear in the blood vessel.
Reduce blood flow?
Exactly. It's going to reduce blood flow to the region. You're constricting the vessels. There's a, basically there's a breach in the wall. So let's like reduce the flow into the region so we don't have as much leaving. Next, we have what's called the platelet plug forming. So platelet plug. So let's say here's my blood vessel. And let's say here's my rip. As the little platelets start activating, they become kind of sticky and spiky. And they start sticking to each other and make kind of like and they actually start release, again, another molecule that has many roles. They release ADP, adenosine diphosphate, which attracts more platelets to the region. So we start getting platelets starting, this is going to be like one of these positive feedback things. They start getting sticky and they release this signaling molecule, in this case ADP, that attracts even more platelets, and they all kind of like sandbag the region. So, and that's going to help keep the blood from leaking out from that region. They form this kind of platelet plug.
All right, you said it's ADP? Okay.
The next thing is we're going to start what's called clot formation. So this is a lot of different steps. Let me make sure I can tell you exactly how many steps. Um, 30 different steps. So in reality, using all sorts of different plasma proteins, but we're just going to kind of put it in a nutshell. So there's a plasma protein that you are going to need to use for this. One is prothrombin. One is called fibrinogen. So these are just kind of in the plasma. Normally, they're not, and they're soluble, right? And they're just kind of dissolved in the plasma, zipping around. During clot formation, there are a bunch of different steps, but the final step is this prothrombin, which has been there all along, gets converted into thrombin. Oh my God. And thrombin is going to catalyze the conversion of fibrinogen into fibrin. And this is insoluble. This is this sticky, fibery stuff that does not stay dissolved in the plasma. And this is going to become the clot, which is kind of like a Band-Aid that forms to kind of seal up the hole. So if we kind of go back over to here, First, we had our little platelet plug forming here. But the next step is we're going to actually get the fibrin. And we're going to actually be able to, you'll see a picture of fibrin on the video. Normally you'd actually see, as you watch your blood clot, you can see it go from being liquid to actually having this kind of stringy stuff in it. So as the blood clots, it starts getting this fibrin happening. So this was like part 3 was the fibrin. Part 2 was the, so at this point you kind of have like a Band-Aid over this thing. Obviously in the big picture, you want to actually heal the damage. So that's going to be part 4. We call clot retraction and repair. So clot retraction, there's actually, I think it's actually actin and myosin. You basically kind of pull the edges together, squeeze all the stuff out. And you actually have, it's called PDGF, platelet-derived growth factor. Let me just write it out. It's going to be easier. And that is basically going to cause the cells, the endothelium and the tunica media to all repair themselves. So this is going to repair the, the wall of the vessel. So this promotes repair of tissue. And so at this point, we now have our blood vessel. We still had that kind of fibrin, but now the wall is all healed up. So then we can actually dissolve the clot. And to do that, there is another enzyme called plasmin. So we have plasminogen, which is just a plasma protein, becomes plasmin. And plasmin is actually the stuff that dissolves the clot. So those are kind of the different steps. In fact, the very first biotech drug ever approved for actual use was TPA, which was made, I think it's still made by Genentech. Does anybody know what TPA does or what it means?
It dissolves strokes.
Yeah, so it dissolves clots for strokes. So a lot of times the way strokes happen is you have one of these, in fact, let me give you a couple of names. The thrombus, that is the word we use. If I have some blood vessels, if I have a clot forming where it shouldn't be, that's called a thrombus. It can happen for a number of reasons, but let's say it happens. If it breaks free, anything that breaks free and is flowing through is then now an embolus. If this thrombus becomes an embolus and breaks free, it might be able to flow here, but eventually it's going to hit some smaller branch and it's going to clog it. and then everything downstream is blocked. So this is a way you can have a stroke or a heart attack, is one of these embolized thrombuses. And so what this drug does, TPA, it's tissue plasminogen activator. So it activates the plasminogen into plasmin. Plasmin then, its job is to dissolve clots. So then you have the plasmin comes in and dissolves a clot and you restore blood flow. So that is only going to work though if you get them in quick. I think there's a window of like, I think 2 or 3 or few hours. If somebody has a stroke and you can get them in and administer this really quickly, there's a good chance you're gonna avoid some of the more extreme kind of, negative consequences of impaired blood flow to the brain. So embolus, thrombus. Back to this picture. Another really common thing people talk about with diseases is hemophilia. I was mentioning that there are like 30 different steps here. If you have some genetic problem with any of the plasma proteins involved with that, that's going to affect your ability to have blood clots forming. And that's hemophilia. So that means it's harder for your blood to stop bleeding once you're cut or once there's some damage. So hemophilia is just some genetic condition where you have some problems with some of these plasma proteins involved in this whole cascade of forming the clot. Finally, I will mention the idea of blood thinners. So they are not really blood thinners, they're actually anticoagulants. Gosh. That inhibit clot formation. So when people talk about blood thinners, what they actually mean is they are taking a drug that inhibits clot formation. Things like heparin does this, Coumadin. Coumadin is also called warfarin. And there's some other ones. So these are drugs that help keep the blood from clotting. And if you're going to take these, if there is an increased chance that you're going to end up with a thrombus, right? So if you're, for whatever reason, you're worried that this is likely to happen, and if that happens, it could give you a stroke or a heart attack by breaking free, you give somebody a drug that makes their blood a little less likely to go there and less likely to get a stroke. Obviously, you have to be really careful titrating your dose. If you give somebody too high a dose of these blood thinners, what's going to happen? Well, somebody.
Is it that not enough blood can travel around the body, like to the brain? Can you start fainting? Or
Okay, so this is where it's kind of tricky for me here because I don't know how much people are following. So it has nothing to do with that actually, right? Right. So again, what do these drugs do? They inhibit clot formation. So if you're Coagul. Gosh darn, I misspelled this, didn't I? Whatever. What happens if you inhibit clot formation? What's not going to happen?
And you can't repair.
It's basically going to be the same idea of what I was talking about with hemophilia, right? Hemophilia is if you are not having this process going properly, where you have clot formation happening. So if you have too much of this blood thinner drug, it's the same basic idea. It means that when the blood vessels are damaged, for whatever reason, you're not going to make the clot and you're going to keep leaking. Does that make sense? So, you know, I mean, you can see it. People who have too high doses of their Coumadin, they get all sorts of horrible bruises just from their socks. Just the pressure of their socks on their feet is enough to break lots of little blood vessels that don't repair, and they get all sorts of bleeding into their tissues. So when you're working with these blood thinners, you have to be very careful with the dosage, and you know, too low, and then you might get the clots that are going to give you a stroke, but too high, they're going to end up giving you bruises, and you'll keep bleeding when normally the hemostasis would keep you from bleeding. All right, so are there questions about this, or this? You should know these there's four basic steps here, right? First step is the platelets are going to get activated when they see connective tissue, when they see the collagen. Then they send out the serotonin, the vascular spasms help reduce the flow into the region. Then they make the plug, they start dogpiling around there to kind of keep the blood from leaking out. Then they initiate this whole cascade of events that we call clot formation, where we have fibrin now kind of making a patch over the area. Then there's going to be the clot retraction and repair, where you actually fix the wall and kind of redo the vessel wall. Then you can dissolve the clot and we're good to go again. So I am going to now return to the lab.